All right, everyone. Hello again. Today's lecture is about PCR and gel electrophoresis. I'm coming to you again from my second bedroom slash laboratory, where I actually have all the stuff you need to do this kind of thing. Here's my little fridge that I have here. That is full of DNA samples that I have extracted from different fish specimens over the years. All the different tubes and things that you need to get that job done. Uh, Here's a bunch of fish samples that I collected a few years ago. Basically, when I catch the fish, I preserve them in ethanol, right? It's basically really strong alcohol, 95 to 100% alcohol solution, actually. And what that does is it draws moisture out of the tissues of the fish, and it preserves the DNA. So basically, I can go back later on and cut off a little piece of their fin. And what that allows me to do is then break down the cells inside and release their contents and purify out the DNA from that mixture. Okay, and once I have purified DNA, what I actually have is a copy of the entire genome, multiple copies of the entire genome, because remember, every cell in the body has an entire copy of the genome. Okay, but what you do with it next, usually you're not going to try to analyze the entire genome at once. Usually what you want to do is look at one single gene or a small subset of genes. And in order to get lots of copies of one gene out of a few copies of the entire genome, you need to use a process called PCR. Okay? And it allows you to focus on one small segment of the genome, so one part of one chromosome, and make lots and lots and lots and lots of copies of that one small part. Okay, it stands for polymerase chain reaction. The P refers to DNA polymerase, which we'll talk about in a few minutes, and then you'll see what I mean by chain reaction later on. All right, so the purpose of this, like I mentioned, is to make lots of copies, thousands of copies of only one small section of the genome. Okay, normally when DNA is copied, and this is something we studied called DNA replication, it happens through the use of various enzymes. Okay, remember, throughout a eukaryotic genome like yours, there are multiple origins of replication on every chromosome. Wherever there's an origin of replication, RNA primase and DNA polymerase attach. They separate the two strands. RNA primase has to create a short primer made out of RNA, right? A little stretch of RNA that is complementary to the exposed nucleotides at the origins of replication. Okay, and then once that primer exists, DNA polymerase can attach new free nucleotides to it in order to extend the strand, right? You remember this is the replication bubble. There's a fork at either end, and the two forks move away from each other as the bubble expands until all the bubbles fuse together, and you have two copies of a chromosome instead of one. Okay, also remember that because of the way the process works, DNA polymerase can only add nucleotides to the 3' end of an existing strand. So you have a leading strand and a lagging strand. The lagging strand is made up of Okazaki fragments, okay, which are made as separate small pieces, and then they have to be joined back together again using DNA ligase, right, which makes phosphodiester bonds between those separate pieces. Okay, so PCR basically uses these ideas and it uses DNA polymerase, but instead of using RNA primase, it's going to use temperature changes to achieve some of the same goals. Okay, so temperature changes are going to help us expose the nucleotides so that we can use them as a template. And then actually, the person designing the PCR would have had to design what are called primers. Okay, instead of being made out of RNA by RNA primase, though, these primers are made out of DNA. Okay, and they help target the process towards the section of the DNA that we're interested in. Okay, so you have to know something about your target DNA first so that you can design the right primers. Okay, because there is no RNA primase involved with PCR. Okay, so DNA polymerase, remember, can't start a new strand on its own. So you have to provide these DNA primers that basically tell the process where to start so that DNA polymerase can build off of them. Okay, and then the last thing you need is free nucleotides as raw materials to build the new complementary strands out of. All right, so like I said, instead of enzymes separating the two strands here, it's going to be temperature changes that do that. So basically, what we need to do is denature the double helix of DNA. All right, so these two black strands of DNA, you can see some of the nucleotides in them. They run in opposite directions because they're anti-parallel. Normally, the two strands are attracted to each other through hydrogen bonds between complementary bases. But if you heat it up high enough using a machine called a thermocycler, and the picture on the right shows the one I have at home. Okay, basically, it's just a really fancy machine that can heat up and cool down very, very precisely and very, very quickly. Okay, so the first step is to denature the double helix or separate the two strands of DNA. And to do that, you have to heat it up to 94 degrees Celsius. Okay, that is very, very hot. And when that happens, there's enough movement happening within the DNA molecules that the hydrogen bonds break down and the two strands of DNA separate. Okay, very, very quickly then, the sample is cooled back down to 52 degrees Celsius. Still pretty hot, okay, but it's cool enough that the primers, again, the short stretches of known DNA sequence, can find their complementary sequences and create new hydrogen bonds. Okay, because the primers are a lot shorter than the chromosomes, the primers will attach to the appropriate sites before the two strands of DNA can come back together. Okay, and then the last step is to raise the temperature up to 72 degrees Celsius, and this is the temperature at which the DNA polymerase is going to work. Okay, and it does the same job, or at least one of them, that DNA polymerase does inside of you. It starts from the primers, and it uses free nucleotides in the solution to build a new complementary strand. Okay, so it's going to read the original template, and starting from the 3' end of the primer, is going to build new strands. Okay, but we can't use the DNA polymerase that comes from you to do this, because your DNA polymerase is an enzyme, all of them are, but yours would denature at 72 degrees Celsius. So the sort of big aha moment in the development of this process was the understanding that organisms that normally live at high temperatures have to have polymerases that are stable at high temperatures too. Okay, so there's a bacterium called Thermus aquaticus, and this picture in the middle here shows a geothermal pool, like a hot spring from Yellowstone National Park. Okay, and these bacteria live inside these hot springs. Okay, and all of their proteins had to evolve to be functional at these kinds of temperatures. So the DNA polymerase that we're using here comes from this Thermus aquaticus bacterium so that it can still function at 72 degrees Celsius. Okay, so this is the first round of the PCR. Okay, so this is the first reaction. We've separated the two original strands and we've built complements. Okay, but remember, this is a chain reaction. So this goes on and on and on and repeats itself. So it's going to go all the way back up to 94 degrees Celsius. It's going to separate the strands again, back down to 52 so that the primers can attach again, and then back up to 72 so that they can build new strands. Okay, now you'll notice here that the blue strands, some of them at least, now stop at both primers. Okay, so the blue strands in the middle are only going to be as long as the section of DNA between the two primers that we designed. And over time, you're going to get lots and lots and lots of pieces of DNA that are only the length of the part of the DNA that we're interested in. Okay, so the chain reaction keeps going, usually for like 30 or 35 rounds of this. And by the time you're done, you have thousands of copies of just the section of DNA that you're interested in. Okay, but you have to make sure that you provide enough primers and you provide enough free nucleotides in order to build all of those pieces of DNA out of. Okay, you don't have to provide a lot of polymerases because remember, enzymes don't get destroyed. They can be used over and over and over again. Okay, but you do have to make sure you provide enough primer and enough nucleotides. All right, so once you have all these copies of DNA, what can you do with them? Okay, so one of the things you might want to do is visualize the DNA to make sure your PCR worked. Okay, and to make sure you're getting a piece of DNA that is the size you expected. Okay, so the process that you would go through to do that is called gel electrophoresis. Okay, so the gel part of this is made with a substance called agar. Okay, basically agar is a long chain polysaccharide, so a carbohydrate, a sugar. Okay, it comes from seaweed, and you dissolve it in a very hot solution that contains a lot of ions. Okay, and the ions are there because this solution needs to be able to pass an electrical charge through it. It needs to be able to conduct electricity. Okay, so when this solution is hot, it's all liquid. But in the picture on the top left, it shows you pour that liquid into what's called a casting tray, and kind of like Jell-O, you allow it to solidify. Okay, the difference here, agar again is a polysaccharide, it's a carbohydrate. Jell-O uses a protein called gelatin, but it's the same idea. Okay, and whether you're making a gel, an agar gel for electrophoresis, or you're making Jell-O, what you're doing is you're trapping a liquid solution inside of some kind of mesh. Okay, so inside of a piece of Jell-O, there's still juice, right? Liquid juice. It's just trapped inside of the protein mesh or matrix. Okay, and here the mesh is made up of these polysaccharides, but it's not solid all the way through. Okay, there's still liquid solution between those strands of carbohydrate. And if there's liquid between the strands, that means things can still move through that liquid. Okay, and it's important here that the mesh have sort of a very complicated and complex web, because that creates very small holes and passageways that something could use to move through that mesh. Excuse me. All right. So once you have your gel solidified, you have to put it in one of these gel boxes here. Okay. And then you're going to take your DNA sample and move very small amounts of it into what are called the wells. Okay. So you might be able to see this here in the bottom left. There are little blue rectangles now within that gel. Okay. Those little blue rectangles, that's the liquid that should contain my DNA. And I've placed it inside these wells, which are basically the starting point. They cut down into the gel, okay, so that the DNA samples are at the same level as the gel. Okay, and then when I put a lid on this and attach it to a power supply, it is going to create an electrical current through the surrounding solution. Okay, and again, remember there is solution within the gel. So the electrical current also passes through the gel. Okay, and that electrical current has a negative end, which is near the wells, and a positive end, which is far from the wells. And that's important because DNA has a negative charge. Okay, so opposites attract. So the DNA inside the wells is going to try to move away from the negatively charged side of this and toward the positively charged side. And that means that the DNA is going to enter the gel and start moving through that mesh. Okay? The DNA isn't smart. It doesn't know to like float upwards and get around it to move faster or anything like that. Okay? It's only going to move in a horizontal direction. So that's why we put the DNA down into the wells, so that when we turn on the current and it tries to move away from negative, it has to move into the gel to get away. All right, so as we run the gel, I mentioned the DNA is moving. Okay? So let's go through some logic here. Smaller pieces can navigate the mesh faster. Okay? And distance equals rate times time. Okay, so all of the DNA pieces experience the electrical field for the same amount of time. Okay, so that means that the distance they move is going to be related to their rate. And for us, that means that the size of the piece of DNA is inversely correlated to distance moved through the gel. Okay, the smaller the piece of DNA, the farther it can move, because it's able to move faster through that mesh. Okay, long pieces of DNA have a harder time negotiating their way through the various tunnels and things in that carbohydrate mesh. All right, so once your DNA has run, then you need to be able to visualize it somehow. And there are various different ways you can do that. One of them is to soak the gel in a blue dye, like that picture on the left. Okay, and when you do that, you can visualize the bands of DNA just using regular light. Okay, so you can see there's actually, I started 2 rows of DNA in the gel on the right. The wells where the DNA started are sort of the open rectangles, and then the dark blue bands are the DNA. Okay, on the right it shows a slightly different thing. There are some chemicals you can put inside the gel that will bind to the DNA and actually fluoresce when exposed to UV light. So that's what you're seeing in the example on the right. Okay, that's a little more dangerous, just because UV light can be dangerous, and some of the chemicals that do this are known to cause cancer. Okay, so we often use the version on the left, but you can do either one. Okay, and they have newer dyes now that are not as dangerous for the fluorescing as well. All right, the last part of this, or the next part, I guess, I'm just going to give you an example of something you could use this for. Okay, so A few years ago, a student came to me trying to find ideas for science fair, and the student told me that they were interested in working with restriction enzymes. Okay, they wanted to find some kind of project where they could use restriction enzymes to cut DNA and figure something out. Okay, so I reached back into my time as a fish person, right? My time as a PhD student, and thought about projects that might be appropriate and actually doable. Okay, and one of the fish I studied was called the orange throat darter. It's a little thing that's colorful. There's some pictures later on. And one of the things that was interesting about orange throat darters that I discovered during my PhD was that even though they are pretty widespread in Illinois, and everybody always thought that all of the different populations in Illinois were basically the same thing, I was able to show that the fish in Illinois now actually came here from two different places. Okay, so in the map on the right, the parts that are covered with gray show all the places that were underneath glaciers during the last ice age. Okay, so there were no fish here underneath the glaciers. Okay, and as the glaciers retreated, fish moved from farther south as they expanded their ranges into these new territories. This particular species invaded Illinois both from the Ozarks in Missouri, that's the purple, and from the Appalachians in Kentucky, that's the blue. Okay, most of Illinois was invaded from Missouri, okay, the Ozarks, but a small part of eastern Illinois was invaded by the population that came from the Appalachians. Okay, and so eastern Illinois, it turns out, is a really interesting area for this. Sort of southeast of Chicago was an area that I thought might be interesting. Unfortunately, this species isn't very common there. But then down around Champaign is another area where these two different types of orange throat darter come together, and they are a lot more common there. Okay, so the data I had collected in order to build this phylogeny during my PhD was from the ND2 gene, which is part of the mitochondrial genome. So I had a lot of DNA sequences from this gene from different populations throughout the range of orange throat darters. Okay, all the little colored dots on the map show somewhere that I went and caught specimens and sequenced DNA from them. Okay, and I had some from Illinois, and I knew that I could find both blue and purple versions of this. So both the blue is the Appalachian and the purple is the Ozark. I could find both of them in Illinois. I had already. Okay, but I was interested to find out where the border was between the two, or if there even was a border, because one of the interesting possibilities was that the two different types, the Ozark and the Appalachian, might be mixing back together, right? They might be hybridizing in eastern Illinois. Okay, so the sequences, the ND2 sequences from these two clades were very different, right? Were different enough that I could look at them and tell whether it was from the blue clade or the purple clade. So I looked at them a little more to see if I could find restriction sites that were unique to only one or the other. And it turned out that I was able to find a restriction site that would cut the blue clade but not the purple, and a different one that would cut purple but not blue. Okay, so what I did next, I went out into the field for a couple weekends, drove between Chicago and Champaign and a little farther south of Champaign, and collected a bunch of fish. Okay, so these are all males. They're kind of like birds, where the males are really colorful and pretty during the breeding season, but the females aren't really. Okay, but different species have different color patterns and stuff. This is one of the prettier ones. I brought these back. The student was able to extract the DNA from them, and then the student was able to perform PCR in order to get lots and lots of copies of the ND2 gene. Okay, and then once she had all those copies of the ND2 gene from the various specimens, then she exposed those copies to restriction enzymes to see if they would cut them or not. So here's an example of the kind of data that we got. You can see, so in the top row there, the first two samples on the left came from the same specimen. Okay, so on the left, we tried to cut it with the restriction enzyme from the blue clade, and then the second one in, we tried to cut with the restriction enzyme from the purple clade. Okay, so in this example, the piece of DNA in the second column represents the complete ND2 gene. And the two pieces of DNA in the left column, the first column, represent the two pieces that were left over after the whole thing was cut by the restriction enzyme. So that told us, because the blue enzyme could cut it, but the purple one couldn't, that told us that the DNA sequence of that ND2 gene would have put this particular specimen in the blue clade. Okay, and so in this picture here, the yellow star shows where Champaign-Urbana is. Okay, and then the shaded purple and blue and the dots inside of them represent the known distribution of this species in Illinois, and my idea of where the purple and blue clades were based on my previous data. Okay, and then the colored dots between those two shaded areas, those are the locations where I collected new specimens for this study. Okay, so you can see at the bottom, the south, there was a place where it was only purple. Okay, as you move up to the middle, though, there's one where there's three colors, right? Purple, blue, and yellow. The yellow means that it was actually a hybrid with a whole other species. We don't need to get into that. But at the very top in the north and that one towards the south, we found both purple and blue alleles in the same place. Okay, so that was really interesting to us, and those were the results of her study. So that was a way to use PCR and gel electrophoresis and restriction enzymes to come up with an answer to a question. Okay, so that's where her project ended. I did a little more after that because I just wanted to double check that the restriction enzymes were doing the job that they were supposed to do. So I actually sequenced DNA from some of the same samples, using some connections at the Field Museum. And so let's just talk a little bit about how DNA sequencing works, because it's related to PCR. Okay? All right, so here's a piece of our DNA that was created by PCR. Okay? So these sections of DNA, again, they start and stop at the primers. All right? So using the same ideas as with PCR, mainly using temperature in order to separate the strands, using Thermus aquaticus DNA polymerase in order to build new strands at a high temperature. Those are all the same with this kind of DNA sequencing. Okay, and you still need primers and everything, right? The same primers as the first time. The only difference is the kinds of free nucleotides that you make available to DNA polymerase. Okay, instead of all of them being normal nucleotides, a small percentage of those nucleotides are modified. Okay, and whenever a modified version of a nucleotide is added to the strand stops being built right there. Okay, so that's one thing that's special about them. Basically, they don't have a 3' end. Okay, and if they don't have a 3' end, then you can't attach a new nucleotide to it. So that's how they make sure to stop there. Also, though, they have a molecule attached to them that when you hit them with a laser, it fluoresces a specific color. Okay, so all the cytosines fluoresce a certain color, all the adenines fluoresce a different color, and so on. And because each of these ends up being a different length, there's a special machine that you can run them through that is kind of like gel electrophoresis. And the shorter the piece is, the faster it moves. Okay, so basically what happens is you start all the DNA off at one end. There's a laser at the other end. And you expose the DNA to an electrical current. So the DNA moves through this really long sort of liquid gel thing. And by the time it gets to the other end where the laser is, the pieces of DNA have separated out by size. So the shortest ones are going to go past first, and then they basically line up along the way. So a piece of DNA that's 20 base pairs long passes before the laser before one that's 21, and then 22 and 23. And every time it passes by the laser, the nucleotide where it stopped being built fluoresces with some kind of color. And then the machine can tell what color it is, and then based on that, the machine can tell you what the order of nucleotides in the sequence is. Okay? If the piece of DNA that is 25 bases long fluoresces green, then base number 25 is a T. Okay? That's how DNA sequencing works. All right, well, hopefully that will help you understand the other stuff I'm going to assign. And that's the end. Bye.